Hi, welcome everyone. I'm Dr. Zhong. I'm your host for today's Bring Health at You Lunchtime Forum, organized and hosted by the Department of Brain Health and UNLV. So today's topic is Alzheimer's, what's new in prevention, diagnosis, and treatment. It's such an exciting time we live in right now because there have just been so many breakthroughs are really shaping our diagnosis, treatment, and Alzheimer's. And today, I have two great speakers here. We have Dr. Jeffrey Cummings, the very well-known neurologist and Alzheimer's expert. He is also the director of the Chambers-Grundy Center of Transformative Neuroscience and UNLV. We have Dr. Jefferson Kinney, who is the chair of the Department of Brain Health. Also, he is the director of the Palm Cork Brain Health and Biomarker Lab at UNLV. So we have two Dr. Jeffs here today to share with you the update, the exciting news about Alzheimer's. We are open to the questions you may have during the discussion, and feel free to use the chat function of the Zoom that we will try our best to get to each and every single questions. Towards the end of the session, there is also an email that we will show. You can feel free to send the questions through the email, as well as any future subject that you might want to discuss and share with the team. And we kicked off this series in September last year. So in the past almost 12 months, we have covered various topics that is very important to bring health, from the benefit of physical exercise, nutrition, sleep, the connection between diabetes and brain health, women's brain health, and among other topics. So today we will be focusing specifically on the advancement about Alzheimer's and brain health. So on that note, I am going to turn first to Dr. Jeffrey Cummings to just tell us a little bit about this breakthrough news that we heard about in the last couple of weeks. And I think that the whole Alzheimer's world has been very excited about this.
Thank you, Kate. And it really is an unbelievably exciting time in Alzheimer's disease. Why is that? It's because we have not had therapies that slow the progress of the disease. We've had some therapies approved 20 years ago that temporarily improve symptoms. They're very important. We continue to use them, but we've never been able to actually modify the biology of Alzheimer's disease to slow its progression. And now it looks like that has been done. So there was a drug approved last year by the FDA called aducanumab, or Aduhelm, and that drug slows Alzheimer's disease, but the trial to show that was less convincing than the recently published trial. So about 2 weeks ago, as you said, a new trial by a very similar drug called lecanemab, and it doesn't have a trade name yet.
Jeff, can you spell that for our audience?
Yes, Lecanemab. So MAB at the end of the name means antibody, AB, antibody. So we can always tell that. So lecanemab unequivocally slowed the progression of Alzheimer's disease. And it also had a dramatic effect on reducing the amyloid plaques in the brain of Alzheimer's disease patients. So the plaques came down and the disease was slowed. And we have never seen that in Alzheimer's disease before. So we're very excited about this. This drug, of course, then has to be presented to the FDA. The FDA has to review the evidence and hopefully is convinced by it. I think they will be, given the strength of this evidence, and then it would be approved. In addition, we have another plaque-lowering antibody that will be announced within the next few weeks. We know that it will be announced before the beginning of December. So within the next few weeks, we're going to see results on another remarkable drug, and we'll see whether it looks as good as the results of the lecanemab study were. But this is a turning point, Kate, for Alzheimer's disease research, and I think for brain research in general. It's been very difficult to actually slow the progress of diseases that kill brain cells. And we don't have such a drug for Parkinson's disease. So we don't have such a drug for Huntington's disease. So these are other forms of cell death diseases. And now we have hope that the learnings from Alzheimer's disease could be extended to them. And I just want to make one, go ahead. Go ahead. I want to make one point that to bring Jefferson Kinney into the discussion is the reason that we know that we're actually modifying the biology is that we can measure aspects of the brain disease either with brain scanning or with blood tests. And with both of those then called biomarkers, then we can see that we are actually affecting the biology of the disease. And Dr. Kinney is an expert in these biomarkers and might want to add to this.
Yeah, thank you, Jeff. I will second that this is a really exciting time in this because there hasn't been treatments evolving at the rate that they are now. And it's remarkably interesting, but it's also remarkably positive for what we're able to do with these diseases. But I also wanted to, as Jeff mentioned, talk a little bit about the biomarkers here, because what they actually provide is the opportunity to really start to understand what these treatments are capable of doing. So included in these trials is the evaluation of whether or not lecanemab is in fact altering the pathology in the disease. So it's not just about an evaluation of somebody reports that they're doing better. We have the ability to really determine whether or not the drug is engaging the target, if it's doing something to the disease process, which is the really exciting data out of all of this, is that we know that one of the core pathologies of Alzheimer's disease is decreasing due to this treatment. And that's such a remarkable advancement compared to where we have been for the last 20 years. So the general way that I oftentimes talk about biomarkers is to make sure people are a little more familiar with them, right? Because when you say biomarker, people don't necessarily always jump onto what it may mean. So the analogy that I found useful is all of us remember the stories about Paul Revere riding along saying the British are coming in the Revolutionary War. And in some ways, that analogy holds true for what we're looking at for biomarkers. They're really just reporters that we can detect a disease in some way, shape, or form, whether it be from a scan, as Jeff highlighted, or from blood samples, which is primarily what we do. And this background actually behind me is our biomarker lab that we've built over the last several years. But the idea is to look for proteins, and a protein is really just something that has an activity. So cholesterol is a protein. You go to your physician, they look at your cholesterol levels, and it becomes a marker that is useful to understand whether or not a disease may be present or whether or not a disease may be coming. And the goal in all of this work, especially in Alzheimer's disease, is to identify proteins, biomarkers, that indicate that a disease is there. Ideally, we work towards identifying whether or not a disease is coming. But the other great phenomenon that happens with this is if you identify something that lets you know a disease is there and you can measure it, it also has a way of being able to evaluate the amount or the severity. And on top of that, as Jeff highlighted for this really, really promising data in 2 weeks ago, is that this can be used to evaluate whether or not a treatment is doing what we want it to do. And in lecanemab, that's precisely it, is that we know that the treatment is reducing amyloid. And that's the first and earliest pathology in the disease. It is the one that we want to engage. And we have a number of really promising biomarkers that are now in these clinical trials. but also in our research to understand the disease. And the goal, of course, is to detect disease, be able to predict disease, and we're making tremendous progress in that space every single year.
That's truly very exciting, isn't it? So Jeffrey, you talked about just in the last year, we had 2 already new treatment. One is called Adu, one is lecanemab, and both drugs reduces the amyloid, which is really what Dr. Jefferson Kinney was just talking about as a biomarker. So I think it sounds like then the connection between the lab, the biomarker, and also the clinical significance is so critical for us to not only understanding the disease, as well as understand if the treatment is working or how well the treatment is working. So I wonder, Jeff, if you can come in, so if you can talk a little bit about how amyloid as a biomarker is currently being used in the clinical setting to help doctors and providers to diagnose Alzheimer's.
Yes. And let me say a little bit, Kate, about how we think this works. I think our audience knows about the immune system. We've all learned a lot about that with COVID, that when you're infected, you generate antibodies to attack the virus and remove it from your body. So these antibodies recognize that there's an abnormal protein, as Jeff said, in the body, and that might be a viral protein, such as COVID, or it might be a protein that is associated with Alzheimer's disease. So in these drugs, the antibodies are made externally in a factory, and then they are infused intravenously into the patient. And those antibodies know that they are looking for the amyloid protein. And the amyloid protein has been associated with the cause of Alzheimer's disease. So these proteins are acting a little bit, these antibodies are acting a little bit like what we're seeing in COVID. They're going after the foreign substance that is in the brain. So this is a really remarkable technology where we train the immune system to do what we want. And we might be able eventually to take the next step, which would be a vaccine. So rather than having to have an intravenous treatment, you would just get a vaccine, much like polio vaccine, and it would prevent you from getting Alzheimer's disease. Wouldn't that be great? So we're making tremendous progress, but these successes are based on the immune system. And we have a scan of this protein. So you can do a brain scan. And before you do the scan, you give a chemical in the vein, and that chemical will only attach to the amyloid that's in the brain. So then you scan it with a scanner that can see that chemical. So if the chemical is hung up in the brain, it's because it is attached to amyloid, and that person has at least the beginnings of Alzheimer's disease. Now, these scans are approved for general use, but they are very expensive, and they are not paid for by any insurance or by Medicare. So they would cost somewhere in the range of $3,500 to $6,000 to have such a scan. So they are used very extensively in research and very little in clinical care.
So Jeff, if individual wish to have this kind of a scan done here in Las Vegas, where can they go? Or is there a place they can get this done here?
Yes, so it is done in the Cleveland Clinic. It is also done at Desert Radiology, and it may well be done in Steinberg. I don't know that for a fact. So just to speak to the local vendors, And if I excluded someone who's a local vendor, I apologize. I'm not trying to promote any special vendor here. But of course, you and I both worked in Cleveland Clinic. We did a lot of clinical trials there, and we did a lot of these scans in order to decide whether the patient was appropriate for a clinical trial or not. But those were research scans, and research scans are always paid for by the researcher, not by the individual. Whereas if you go just for a diagnostic scan because you want to know whether the protein is there, then that person would have to bear the $3,500 to $6,000 cost of the scan. Now, we're really hoping that Medicare is going to start paying for those scans. And with the success of the drugs, such as this lecanemab that we've been talking about, it's very likely that they will, because those drugs should never be given to somebody who does not have that protein in the brain, because then you could have the side effects and you couldn't have the benefit. So you must know that the protein is in the brain. And the only way to know that for sure right now is with the scan. So I think it's pretty likely that as this goes forward, and we're just in this new era, how great is it? We've turned the corner, we're in a new era. When we're in this new era, I think Medicare is going to have to re-examine their strategies for taking care of older patients with Alzheimer's disease.
So just reiterate that for every single patient who was recruited for the clinical trial for the lecanemab, they have all gone through the confirmatory diagnostic process using the amyloid to confirm they actually had the amyloid protein present in their brain before they became eligible, among many other criteria, to enter in this trial. Is that correct?
That is correct. And it also brings up another point, Kate, if I could, which is this trial included people with very mild memory impairment, not severe disease, very mild disease. Well, a lot of people with mild memory impairment do not have Alzheimer's disease. So they would have a $3,000 scan and it would be negative and they couldn't participate in the trial. So to get the 2,000 patients that participated in that trial, all of whom had positive amyloid imaging, they would have had to do, and I don't know the exact number, but on the order of 10,000 scans, because the failure rate or the rate of non-detection of amyloid is quite high. So you can see the expense of developing a new drug when you have to do 10,000 scans, and most of them result in not including the patient in the trial.
That really speaks to the importance of having more people involved in clinical trial, isn't it?
Yes, very much so.
A question from Irene: are there any major risk of this new medication, and are there any limitations on who can take them?
Yes, so really great questions. And this raises one of the great controversies of the use of this particular therapy. And that has to do with something called amyloid-related, we've been talking about amyloid, imaging. So these are seen on MRI abnormalities, amyloid-related imaging abnormalities, or ARIA. And ARIA occurs because fluid leaks out of the blood vessel into the brain. And sometimes blood itself will leak out of the blood vessel into the brain when you give these drugs. Why is that? Because there is this same protein, amyloid, that we're talking about, that causes Alzheimer's, is also in the blood vessel. So as the antibody starts to pull the amyloid out of the brain, it also pulls the amyloid out of the blood vessel and temporarily makes it possible for fluid to escape into the brain. That happens in about 40% of patients who participate in this therapy. However, less than 5% will actually have any symptoms that allow them to know that this has happened. So we scan patients with MRI because we want to know if it has happened. We might even interrupt the therapy temporarily if it looks like it's substantial. After 1 year, those blood vessels heal, and it's very rare to have the ARIA after 1 year of therapy. But it is a complication, and some doctors have concluded that they will not give this because they believe that the potential for harm exceeds the potential for benefit. I personally do not agree with that. Most of these patients, a huge majority, have no symptoms. Occasionally, symptoms are severe. But of course, Alzheimer's is severe. It's a fatal illness. So if we can slow Alzheimer's disease, to me, it seems that it's worth the risk of this ARIA. But I truly believe that that is every patient's decision, not my decision. That is every patient's decision. So we have to tell the patient about ARIA, and then we have to help them make the decision whether the benefit and the harm are in balance for them. Now, just briefly, Kate, address the other question that was raised, which is, are there any limitations on who can have it? Well, the person has to have memory impairment that is not severe. They cannot be in the severe stage of Alzheimer's disease. And they have to have—
Jeff, can you just clarify when we define severe? So for our audience, how do we judge? If, mild, moderate, and severe. So we have the whole spectrum of the severity of the dementia in the Alzheimer's patient. How do we define those?
Yes, really great question. And the patient would be helped by their doctor in deciding whether they had mild or moderate or severe disease. But in general, you would say that somebody who has a lot of functional disability, they're getting lost readily. They're forgetting the names of their relatives or their spouses. Those things those are things that we consider characteristic of more severe disease. So a person would not have that. They would be forgetful, but they would be largely functional. They could still probably do their shopping. They could still do maybe their own driving. So they're in that very mild phase. So they have to have that. And as we've said, they have to have a positive amyloid scan. And for most of the drugs, they have to be, well, there's only one available, aducanumab. For that drug, you cannot be on an anticoagulant or blood thinner and still get the disease, get the drug. So there are some requirements but they're relatively limited requirements if the patient truly has mild changes.
Thank you. So another question before I move on from Richard, I think it's great. Richard mentioned this with regard to the FDA's approval requirements. So Richard asked, FDA has been reluctant to consider endpoint for clinical trial before observable physical indications of actual symptom of a disease. So will the FDA allow the trial endpoint using biomarker alone instead of waiting for the clinical indication that take much longer, take much more time? So I think it might be helpful for Jeff, you can just share with our audience in terms of the endpoint used for this clinical trial, both from the biomarker perspective as well as from the clinical observation perspective.
Yes, so that's a really great question, Richard. And it has also been the source of controversy. Whenever there's progress and change, there's also controversy. So I'll establish a background by saying that it is common in cancer for a drug to be approved if it reduces the size of the cancer. Well, that's a biomarker, right? That's a biomarker. But in general, in Alzheimer's disease, biomarkers were not powerful enough to be the basis for approval of a drug. So we had never had an approval of an Alzheimer drug based on a biomarker. But when aducanumab, the currently approved drug, was presented to the FDA, they were so impressed with the amount of removal of protein from the brain, the toxic protein, that they approved it based on the scan results. When they do that, the company also is told that they must conduct a confirmatory trial which shows the clinical benefit. So what this allows is that patients can have access to very promising therapies before they are completely proven to be a benefit. And if in the confirmatory trial you don't see the benefit, the drug is removed from the market. But if we had to wait for a confirmatory clinical effect for every drug, we would have many patients dying of cancer, and we would also have many patients now progressing in their Alzheimer's disease. So the accelerated approval on the basis of a biomarker is a tool that the FDA uses in order to allow patients to have access to drugs before the data are completely compelling.
Thank you, Jeff. Just very quickly, I think for the for our audience, can you share with us in terms of what's the current trajectory with regard to the submission to FDA and potentially the time when the drug, the new lecanemab, potentially can become available for our patients?
Yes, thank you. And I'd like to make one point that came to mind, Kate, which is that although aducanumab was approved by the FDA, the Medicare decided not to reimburse it. And that was a great disappointment to all of us who work in the field and who have worked so hard to develop new drugs, and then realize our patients weren't going to be able to get it unless they could afford out of pocket the $25,000 per year that aducanumab costs. So we have a lot of advocacy going on to try to change the mind of Medicare. But in terms of the time frame, on January 6th, this is very precise, on January 6th, we will hear whether this new drug gets accelerated approval based on its dramatic reduction of amyloid in the brain. And the company who developed this, Eisai, will then give all of the new clinical data to the FDA. It takes many months, I expect 8 to 12 months, for the FDA to review that and make a decision, which would then mean that the new drug would be available and with proven clinical data that we've now seen by early 2024.
It's truly a very long process, isn't it?
Yes.
So, well, thank you for that. Anyone who has questions, please feel free to just send in the chat box. We have so far really spent quite a bit of time talking about amyloid, talking about the biomarker, specifically based on the brain scanning. And as we know that the amyloid can be present in the brain years, even decades before the patient develops symptoms. And amyloid, as you heard, the scanning is very expensive, can be as high as $6,000. And it's not very convenient. You have to go through the imaging and you have to get an injection, so on and so forth. So there really has been a lot of effort and advance made in the other kind of biomarker, right? We call that a fluid biomarker. So I'm going to turn that to Dr. Kinney and to share with us a little bit, in addition to the brain scan biomarker. So what other biomarkers right now are really making really great wave in terms of our understanding for Alzheimer's disease, and potentially what can be a much less expensive and more convenient way for people to get the diagnosis?
Yeah, thank you, Kate. And I think it's a really intriguing and exciting time, not just because of the progress in these trials, but because of the advances that have been made, especially in the last 5 years, in the biomarkers of this disease. So as Jeff and Kate sort of highlighted already, the amyloid pathology in Alzheimer's disease shows up years before clinically they present in a way. So it's already there and increasing prior to typically a clinical diagnosis. And this and the need for these in clinical trials to determine whether or not a drug is actually successfully engaging the disease has driven a lot of progress in the biomarkers. Where it gets a little bit interesting, so the scans that Jeff was talking about are exceedingly useful, but it's a little bit more difficult to look for neurodegenerative disease markers in blood because the brain is a very protected space. It's its own version of a clean room, and not everything that's in your periphery goes in and out of where your brain is. So a lot of the biomarkers that have developed, starting with the imaging ones and now the fluid biomarkers that are detectable from blood, it's a difficult exercise to determine what may be leaking out of the brain that is detectable in blood that lets you know the disease is there, let alone whether or not a treatment is engaging the pathology and improving the pathology in the disease. So the good news is there's been an enormous amount of progress on lots of different markers. There are a couple of amyloid markers from blood that are showing promise, but they're not quite there. And largely this is precisely for what Jeff highlighted, is that amyloid is also in blood vessels. And this makes it a little bit more difficult to separate out what is amyloid accumulation due to Alzheimer's disease and what is amyloid from blood vessels. But there are two very promising ones from two separate companies that have found a reduction in amyloid in blood is associated with increased amyloid in brain. And it's probably worth mentioning, a lot of what we're doing right now in these fluid biomarkers is using both clinical measures, but also the imaging data to evaluate how well the blood-based biomarkers are mapping onto the disease. So the amyloid is a good example. There's two others that we're working on in our lab, as well as a lot of other labs. One is a marker for a protein called tau, and this is the second core pathology of Alzheimer's disease. So the first are these amyloid plaques that aggregate between cells in the brain. And what comes later in the disease is a protein called tau, which in essence, what tau does is it helps hold the structure of cells together. And normally it has to be removed from its spot so that a cell event can happen and then it's returned. And what happens in Alzheimer's disease is this tau protein starts to aggregate together and come off of where it's holding the structure together. And that starts to clump within cells that is also tied to neuronal loss, to loss of neurons in the disease. There is a particularly promising tau marker. It's called p-tau181, and that just simply refers to a specific position on the protein. But from blood, we are seeing a really strong association between this marker and amyloid volume from scans. And in fact, our group has a publication that we're about to send out that really is one of a few highlighting this association. One of the other ones that's really showing a great deal of promise is something called neurofilament light. It's a marker that is basically, it's an indication that neurons are dying. So it's a protein that should be inside neurons, and if neurons are dying, it makes its way into the bloodstream. So if levels of it are increasing, it's an indication that you may actually be losing some neurons. And the last one I think worth mentioning, because Jeff talked about utilizing the immune system to engage the disease. One of the other real central tenets of the disease for the last 10 to 15 years has been there's an immune response that is associated with Alzheimer's disease. So as the amyloid starts, the brain's natural immune system begins attacking the amyloid to try and clear it away. But what this results in is an immune response that is sustained over time. And this isn't really how your immune system was developed to react. It's more about attack and then go back into rest. And this sustained immune response actually propagates, it accelerates and even enhances both amyloid and tau pathology. So not only is this a promising area of therapeutic development, it also provides an opportunity to look for immune markers that may indicate there is a response going on in response to the pathology in Alzheimer's disease. And there are a few immune targets from blood that are showing promise, one above all others. And so in this picture, this is my biobank manager, and this is one of my graduate students, and I think they're even on the call, so they're smiling right now. But they'll tell you that I am just over the moon about this glial protein that we're looking at, because in our hands, and from what I understand others, we are seeing remarkable associations between the level of this protein, amyloid load, and we're even trying to map it onto some of the clinical variables. So that I'm really fair and clear about this, right now, the goal is to get these fluid biomarkers to a point that they are really effective to screen, to identify people that are more at risk, as well as to be used in clinical trials. But the goal of this is to develop a blood test assembly of markers that would be prognostic, right? That would be able to determine whether or not someone does have the disease, but in the ultimate version of this, be able to predict the disease onset several years from now.
How exciting that is. And that is really happening right here in Vegas, in our lab in UNLV. So thank you, Dr. Kennis, for sharing those exciting news, and please keep us posted. I think that in the very near future, some very exciting diagnostic assay may come from our own very lab. How cool that will be. So you know that this is really a scientist talking, right? From the microglia to the immune response to the inflammatory process, and so on and so forth. I just thank you for that. I want to touch upon a little bit. You touched upon the blood biomarker and fluid biomarker. What about the CSF? What about spinal tap, spinal fluid? That also has been a very important area with regard to the biomarker, isn't it?
Yeah, no, and thank you for actually, I always jump right to the blood-based ones because it's a lot where we're digging. So cerebral spinal fluid, so taking the solution that's running around inside your nervous system has been particularly productive. So amyloid and tau measures in CSF have been very, very accurate. They're very, very promising. It just goes with the necessity of actually having to collect it, right? And this is not something that a lot of people, think first that they'd like to go to their doctor and have a spinal tap done. But because that's within the nervous system space, it's been remarkably productive and really, really accurate.
Maybe I could make one point to that, Kate, is, of course, because the spinal fluid is much closer to your brain, it actually bathes the brain. There are a lot of chemicals that we can see in the spinal fluid that are simply not detectable in the blood. So the ones that Jeff talked about are just so exciting because we can see them in the blood, like the 181 marker or the neurofilament light marker. Those are both detectable in the blood. That is really great. But there's a whole group of biomarkers that can be seen only in the spinal fluid because they are only detectable. There's enough of them in the spinal fluid, a high enough level of them to be detected by our current technology. Now, what we're hopeful about, and I know Jeff shares this hope with me, is that the sensitivity of our technology will progressively increase so that at least some of these that we can now detect only in spinal fluid will eventually be detectable in the blood. Because the more we can see in the blood, the more we can understand about that individual patient and how to help them.
Yeah, I will absolutely second that statement. In that, just in the last 5 years, the sensitivity of the equipment that we can point to these projects has grown by orders of magnitude, and it continues to do this. So as the tools get stronger, we keep discovering new layers of candidates. NFL is just a few years old. This tau marker I mentioned is only a couple of years old as well, and it's because now we have the capacity to detect them.
Thank you. Just a last question about the research. Richard has a question about the using transgenic mouse. Has that been used in terms of researching the new drug for Alzheimer's, as that has been used for the other diseases?
Yeah, we're both probably going to
with that, and I'll follow up.
Yeah. So another part of the research that we do in our lab is actually on these preclinical models. So the transgenic mouse models of Alzheimer's disease, what these really refer to are mice that have gene mutations that are associated with familial Alzheimer's disease. So this is about 2 to 3% of our Alzheimer's patients, where there are very specific mutations on very specific genes that result in an earlier onset version of Alzheimer's disease. And these have been unbelievably useful to understand the biology of Alzheimer's disease. A lot of the compounds that have been tested in clinical trials were first tested in these transgenic mouse models. But one of the things that the field has really started to pay attention to is evaluating compounds, or really the generation of mouse models, preclinical models, that aren't based on these genetic familial Alzheimer's mechanisms. So they've been really informative, but I think as a whole, and Jeff can certainly speak even more thoroughly on this in terms of the decision into clinical trials, that there is room to improve on what it is that we're doing with the models, but we have used them quite a bit.
And I would just add, Kate, that we have successfully cured Alzheimer's disease in many mice, and that has not translated into human benefit. So the mice often don't produce the kind of predictive data about the human response that we want. On the other hand, if the drug did not affect the amyloid in the mouse, we'd be very reluctant to advance that drug into a human because the mice and the human amyloid is so similar. So it is a kind of gateway mechanism, even though it's not a good predictor of the response that patients will have. So these drugs have been studied in transgenic animals and shown to be a benefit in the transgenic animals.
Thank you for that. So we have talked about the diagnosis, and we touched upon the exciting new treatment. I'd love to move on and just really touch upon a little bit on the prevention, since that we are all very interested in knowing what we can do to prevent Alzheimer's, so we don't get Alzheimer's. Nobody gets Alzheimer's in the very near future. There are obviously many efforts made in this particular area. So Jeff Cummings, I would like to turn that to you first, and then give us a little update on the prevention trial specifically. That is the clinical trial, ongoing clinical trial, or clinical trial that's been currently in the planning phase, specifically looking at how the Alzheimer's disease can be prevented, not just treating or slowing down the progression.
Yes, thank you. And Kate, you made the critical observation just a few minutes ago, which is that the amyloid begins to accumulate in the brain and is at substantial levels for at least 15 years before the patient has the first symptom, before there's any detectable change in their memory. So that creates the opportunity for prevention, right? Because the person is normal. So if we could get rid of the amyloid at that point, we would either greatly delay or completely prevent the occurrence of Alzheimer's disease. That's such an exciting proposition. So the drugs that are showing promise by greatly lowering these amyloid levels are going into prevention trials. So the way those trials are done is that people are cognitively normal, but they have the brain scan and they show that they have the amyloid in the brain. Now, how do we know that, right? They're just normal people. How would we know that they have, why would we want to screen them for amyloid in the brain? It's because most of them have had affected parents. So that's a great indication that that person has some degree of increased risk. There'll still be a lot of people who do not have amyloid in the brain, even though they have affected parents. But there will be many more if they have affected parents that will have the amyloid in the brain. So they're cognitively normal, they have amyloid in the brain, and then they're put into these prevention trials to see whether the emergence of the symptoms of Alzheimer's can either be deferred, maybe by years, or maybe completely prevented if we remove the amyloid. So both lecanemab, the drug that we have exciting news on, and gantenerumab, which is the one that we're going to hear about in the next month, are already in prevention trials. Now, those trials have to be very long, about 5 years, because it takes a long time for a person to slowly develop the symptoms of Alzheimer's disease. So, of course, if you're trying to prevent it, you also have to watch a long time to see whether you prevented the emergence of Alzheimer's disease. So we will not have an answer on this for several years, but it is a really exciting proposition that we could prevent or greatly delay the onset of Alzheimer's disease. It would be a game changer for so many patients who are at risk for this in their late life.
So for those who are interested, particularly those who may have parents or family members who have Alzheimer's and therefore put them at a higher risk, if those people who are interested in participating in prevention trials, how do they find out about the information? Where can they go?
Yes, so I do not think that there are any such trials in Las Vegas, but they can go to Trial Match on the Alzheimer's Association website. And with their area code, they can find out what the nearest trial site would be. So Trial Match is a really great tool for doing exactly what you have asked.
So you have touched upon the prevention trial, specifically focusing on using the pharmacological intervention or drug. I think there are also some other trials ongoing looking at how potentially through engaging in different lifestyles or bring healthy lifestyles can also change the course or to potentially reduce the risk and prevent Alzheimer's. Jeff, I wonder if you can just touch upon that a little bit as well to our audience.
Yes. So, Kate, I'm reluctant to do this because you are the expert in this field. But since you've asked, yes, there's a lot of evidence that the risk of late life cognitive decline can be reduced, and that is done by a combination, a multimodal approach of diet, exercise, and control of cardiovascular risk factors in particular. There are other things like sleep that are also important, but those three, diet, exercise, and cardiovascular risk factors, are particularly potent in terms of reducing the risk of late life disease. And there's something going on around the world right now called the Pointer Study, the worldwide Pointer Study, that is an international study of a multimodal lifestyle intervention. And we're very hopeful about that trial and learning how we all can help reduce our risk for late life cognitive decline.
I think just want to carry on with that discussion that the very prominent study that was done in Finland a few years back, it's called Finger study, that really established the foundation of the overall effectiveness of the multi-domain approach through what Jeff has just touched upon, physical exercise, healthy eating, and brain stimulation, among others. So this World Finger Study, World Pointer Study is a continuing part, adapting the similar approach, but rather than just focusing in few countries, this is becoming a worldwide approach. And in our last, pretty much last year, that through this series, we have really targeted specifically on all those aspects. So we have the link for you to review if you wish to. And I think there are some very interesting and important update and information for your reference specifically on that topic. There's a quick question from Gwen. So Alzheimer's is hereditary. Is there any age limit for the scan? Dr. Cummings, maybe you can answer that question.
Yes, and I see we're running out of time, yeah. So I'll be brief. What is inherited is a risk for Alzheimer's disease. So if one has affected parents, then the risk is increased, but it is not a sort of one-to-one. My dad had Alzheimer's disease, and therefore I'm going to get it. It's more like my dad had Alzheimer's disease, and therefore my risk is increased, usually about 3 times. So if somebody does not have an affected parent, their lifetime risk is about 15% getting Alzheimer's disease. If they have an affected parent, their lifetime risk goes up to around 40%. So it's a substantial increase, but still most of the people, 60%, who have an affected parent will not get Alzheimer's disease. So it's a risk, and in the case that I was discussing, it helps us find patients who are at increased risk and therefore might have a positive scan and could participate in the trial. But it is by no means fate that you will have Alzheimer's if your parent does. It simply means that you need to be more vigilant. Do your exercises, have a great diet, and control your cardiovascular risk factors, because we know that those things work even in people who are genetically predisposed to the illness.
And is there any age limit on getting the scan done?
There isn't any age limit. On the other hand, because it becomes positive about 15 years before symptoms, and the average age of onset of Alzheimer's is 73. So it wouldn't make any sense to get the scan until you are 66 or 65, something like that, because the likelihood that you would have it is just exceedingly small. But you can get it if you want to know whether you're accumulating the protein in the brain and you have the money for the scan, it would make sense to get it at, say, age 65 or older.
Okay. Very quickly, Dr. Kennis, I want to give a shout out to the lab and to our department that we are doing many more project and research. And so there is one that's currently ongoing. I just want to give a shout out to Sam's project. Maybe you can just share with the audience very quickly as to what they potentially can participate.
Yeah, so one of our faculty members is particularly interested in both socioeconomic as well as diversity differences in Alzheimer's disease. So she has a lab working with participants here in Las Vegas. So it's the Cognitive Aging and Neuropsychological Equity Lab, or the CANE Lab at UNLV. So if anyone is interested in learning more about it, you can email those of us on this call, or you can look up the CANE Lab. It's part of a really integrated approach here within the Department of Brain Health, where we leverage our expertise across clinical work as well as some of the biomarker work to really start to address both basic questions in the disease, but also things like cardiovascular health, diabetes, diet, really focused on Alzheimer's disease research.
Thank you, Jeff. We also have Dr. Hakim here. She has a question about the role of the statin in the prevention of Alzheimer's. Jeff, can you comment on that?
Yes, it's been controversial. There was some evidence that statins reduce the likelihood of developing Alzheimer's disease, but that could not be reproduced in trials. It is likely that if one has hypercholesterolemia, that the statins would reduce your risk for stroke and vascular related cognitive decline. But it is not proven, and the trials have been negative for the utility of statins in Alzheimer's disease.
Thank you. So I'm going to wrap up this, and as we heard from our experts, a very exciting time in terms of Alzheimer's prevention, diagnosis, and treatment. And there are so many more to come. Next month in San Francisco, we will be having the International Alzheimer's Clinical Trial Conference, so that there will be more data to be shared and more exciting news yet to come. Thank you all for coming today. I'm going to turn this back to Andy to wrap the session up.
Hi, everybody. Thank you for joining us today. We hope the information that was shared with all of you was informative and helpful. As you can see on the screen, we have a newsletter that you can subscribe to, and you can just open your photo app and scan that QR code to do so, or you can just shoot us an email at brain.health@unlv.edu. We always love to hear from you, your questions, comments, or suggestions. So if you had a burning question that was not addressed today, please feel free to send us an email. We'll be happy to reach out to you. Also, we will follow up with more information regarding the Kane Lab. I did put the trial match URL link in the chat. If you missed that, I will be sure to include that in a follow-up email to all of those who have joined us today. And just be on the lookout for other news. There's always something exciting and new happening coming out of the Department of Brain Health. So if you haven't subscribed to the newsletter, we encourage you to do so. Until we see you again on November 23rd, everybody have a safe and blessed month.
Thank you all. Have a good afternoon.